Hey everybody, thank you so much for clicking on this video. As an AP biology teacher, I truly enjoy each year going through the AP FRQs that are released to the public. So this year we're going to go over it. This is the 2025 AP Biology FRQ test that happened on May 5th, 2025. It's really important for you to know that all the answers in this video are not official. These are my predictions. I took the time after they were released today after school, and I sat down and I did it myself for 90 minutes. And then from there, I created this slide deck for me to kind of best illustrate to you what my guesses are for each of the answers. Now, it's really important to know that the official College Board scoring guidelines and samples and scoring distributions will come out usually in July 2025. So if you're watching this after that time, go ahead and click below, and I'm going to go ahead and leave a link to those as well when they come out. All right. So as I said earlier, timestamps for each of those are going to be below. If you want to navigate to a certain number on the AP FRQ, you can do so. Some of you are watching this video because you already took the exam and you're interested to see what the answers are. Some of you are watching this because you haven't taken the exam and you want to use this as a practice. Regardless of why you're watching this video, I hope this is helpful. And once again, below, after July 2025, I'm going to put a PDF of the answers. But even right now, when this video is posted, I'm going to put a PDF of the actual exam questions below. So if you want to go in and do it on your own first, you can go and do that and then look at these predicted answers. All right, cool. Please note, once again, as I said before, these are not the official answers. These are my predictions, and this is just me having fun and going through the FRQ to better help my teaching as an AP biology teacher. So go easy and comment below if you have any improvements or other answers that you believe that are right. Once again, it's also important to know that the AP FRQ scoring guidelines, when they come out, there's usually multiple answers. So maybe the answer I put is an answer, and maybe the answer you put is going to be an answer. And as I've said in my previous videos, this YouTube channel is not monetized, and there's really no goal for this channel to be monetized. It's just a fun channel for me to post content on. And if it's helpful for you, go ahead and like it if you want, or go ahead and share it with someone else. All right, what's new this year on the AP Bio 2025 exam? Well, different from last year, this year there was a hybrid digital exam. Now what that means is that the multiple choice was on the conducted on the laptop through a Collegeboard Blue Book app, and the FRQs were actually shown on the student's laptop, and students had to handwrite them onto a booklet that was provided by Collegeboard, right? So it's a hybrid this year. In the future, we don't know what it's going to be like, but that's what it was this year for 2025. It's really important to also know that it says on the Collegeboard website for the exam security policies that 2 days after the regular scheduled exam, which is already passed by now, teachers and people may discuss it. So hopefully this video doesn't go against any Collegeboard exam policies. I'm just trying to learn more as I go through the FRQs and share what I've learned with my students and with whoever wants to listen. It's also important to know that, yes, as you can see, the questions are very secure. So here's just some different things that the Collegeboard website talks about. And then the most commonly administered sets of FRQs are posted on the Collegeboard website 2 days after the exam. You may have taken an exam that looks different than this. And if that's so, then it's probably not publicly posted, but only these questions may be discussed by students and teachers. You know, every single year I get questions on timing. And, you know, it's true. Because you only get 90 minutes to answer the 6 FRQs, it makes it a lot more difficult to do well on these FRQs. If you had 2, 3, 4 hours to do so, I'm pretty sure you would do a lot better. So when it comes to timing, I always like to communicate with my students that of the 6 FRQs, roughly the first 2 FRQs are about 50% of the score. And then 3, 4, 5, and 6 are shorter FRQs. And that's also roughly 50% of the score. So, you know, once again, everyone's going to take it differently. However, the standard rule is if you're given 90 minutes for 6 FRQs, and the first 2 are worth 50%, and the last 4 are worth 50%, it's recommended that you roughly look at 45 minutes for each of those 2 parts, 1 and 2, and 3, 4, 5, 6. So if you break it down, if you divide 45 minutes divided by the 2 long FRQs, it's roughly about 22.5 minutes each. For me, it took about 25 minutes on the first FRQ and on the second one as well, leaving me less time on the other ones. So once again, work on the pacing. If you haven't taken the AP FRQ yet, or the AP exam yet for bio, please take a practice test, and please make sure you figure out what is your strategy for timing. Because even when I was taking it for the 90 minutes, it felt rushed. Okay, so if you feel that way, I think everyone feels that way. All right, so keep that in mind. All right, so here we go. The 2025 AP Biology FRQs. Let's begin with the FRQ number 1, which is considered a long FRQ. There's about 7 or 8 parts in this from what I remember, and you're given about 22.5 minutes. So if you roughly do the math on it, it's about 3 minutes per question, which is very, very fast, not including the time that you would take to read the question. All right, so let's get right to it. When you take a look at the first one, it begins with just a normal introductory question. This is what I call a gimme question. This is a question that most students will get right. I hope you get right because it's pretty foundational. All right, so number 1, most proteins that are secreted from a cell must be transported into the ER, the endoplasmic reticulum, either during translation or even after translation. All that it asks you to do is to describe the function of ribosomes. So here's my answer. The function of ribosomes is to make proteins by translating an mRNA strand into a chain of amino acids, which put together will be a polypeptide. All right, so that's number 1. Let's go ahead and read FRQ number 1. It tells you for proteins transported during translation, the process begins in the cytosol and pauses when a specific sequence of amino acids is translated. The translation complex is then transported to the surface of the ER where translation continues. Proteins that are transported after translation are translated entirely in the cytosol, in the cytoplasm, and then transported into the ER. In both instances, the translated proteins enter the ER through a protein channel in the membrane of the ER. My strategy for when I took this during a 90-minute timer is I like to take notes on the side. Once again, you're viewing this on a laptop. So most likely you can circle, maybe you can highlight using the Collegeboard Blue Book app. However, for what I like to do, I like to write down some notes on the side. What I found is that when I write down notes on the side, I don't have to reread the question. So to kind of type in and to show you what I was writing on the side, I just quickly wrote down that, ribosomes form proteins and free ribosomes form proteins for the cell versus bound ribosomes on the rough ER, and depending on where the ribosome is going to form proteins for outside the cell, for destination of secretion of the cell. There's also a lesson on translocation. Now, this isn't the translocation of mutations where chromosomes can go in and switch parts, but it's more so translocation. It's where a signal can enter the ER through a protein complex. So can things go into the ER? Yes, and it's called translocation. All right. Let's go ahead and continue reading FRQ number 1. Researchers studying the 2 types of protein transport identified that the ER membrane protein SR is necessary for transport during translation, while the ER membrane protein SEC62 is necessary for transport after translation. As an AP biology teacher, I can just tell you right now that we don't study the protein SEC62 or the protein SR. So whenever the AP exam gives you these proteins or whatever topic or whatever name, just know that most likely none of us studied it. All you have to do is use the information that they gave you to kind of figure out what it's asking. So I'm going to continue reading here. To investigate which transport mechanism is used for different proteins, researchers first created small interfering RNAs, which are called siRNAs, similar to miRNAs, that reduce the expression of either SR or SEC62. They then treated groups of cells with either the siRNA or the SEC62 siRNA, so either of those, and determine the relative amount of SR and SEC62 protein in each group of cells compared with cells treated with control siRNA. So here's all I kind of jotted down on the side. This is kind of an elongated version. Go ahead and skip ahead if you want to go ahead and take a look at the answers, but I'm going to kind of walk you through how I solved this problem. SR equals necessary for transport during translation. That was stated in the question right there. And SEC62 is necessary for transport after translation. Now that will go ahead and help me in one question later on, but that's just what I wrote down on the side. siRNAs, similar to miRNAs, they actually, what do? If you don't know what they do, if, let's say you took this exam and you're thinking, I have no idea what siRNAs do, you just got to read the question. They reduce the expression and they pretty much turn off the expression of genes. When we look at chapter 18 in our apbiology textbook, we're looking at how eukaryotic genes are regulated. And that's what siRNAs do. They reduce or they silence protein synthesis. That's all you need to know. So the problem scares you with siRNAs, but it tells you really what it is. Then siRNAs and mRNAs can lead to something called RNAi, which is RNA interference, which once again is used in labs to disable specific genes to investigate their function. Right? I'm going to turn back on this here. All right. So siRNAs, here's a picture in our textbook about miRNAs. There's actually no picture in our textbook about siRNAs, but the difference between miRNAs and siRNAs is that siRNAs are double-stranded. But essentially, they do the same thing. I want to kind of walk you through and teach this to you. When you take a look at the picture of miRNA, this miRNA single-stranded is going to go ahead and bind to a target mRNA. From there, what is it going to do? It can do one of two things in this picture that it shows you. It can either degrade mRNA, or it can go ahead and block translation, which down here, I just told you, siRNAs, they're similar to mRNAs in that they turn off the expression of genes. They reduce or silence. They stop, they halt protein synthesis, all right? So hopefully that helps your understanding. So as I continue and I look at this question, and I'm doing this very quickly, but in this video, I'm slowing it down to kind of tell you my thought process. When you look at figure number one, I just quickly look at this graph. I've never seen this graph, but I'm trying to make sense of it. Here I see that the siRNA added to the cells is the control group. You've got the SEC62, and then you've got the siRNA. Once again, we have not learned this in our AP biology class. This is the exam testing if you're able to make sense of this data and analyze and interpret this graph. So what I'm thinking in my head as I'm looking at this graph is that I'm looking at it in SEC62 siRNA. When it's added, as you can see right here, the SR protein content shoots up. And that's way over the control, about 160, roughly 160%. Versus the siRNA, when that's added, the SEC2 protein content shoots up to about 125%. How does that even make sense? It absolutely makes sense based on what I just said earlier. What do siRNAs do? They halt, they reduce, they silence protein synthesis. So if that's happening, you're not going to see, for example, right here, SEC62 siRNA effect. That's why the protein content is going to be less here. For siRNA, that's why the protein content is going to be less there. It all makes sense based on the information given. So now let's go into the answers. And if you want explanation, you can go back to how I just explained. For FRQ number one, for B1, the question is identify the dependent variable. In class, we learned that the dependent variable is the x-axis. Oh, sorry, it's the y-axis, excuse me. And the independent variable is the x-axis. So what is on the y-axis shown here in the graph? Well, I'm just reading right here. It's the relative protein content percent of control. This should be a very quick answer. When I went through it, I quickly looked at the graph and I answered really quick. So if you were to write it out, you would write out something along the lines of the dependent variable, or the y-axis, shows the relative is labeled by the relative protein content percentage of the control. Specifically, the protein content of SEC2 and SR protein, as you can see. For B2, the question asks, justify why the researchers included the control of measuring the relative amounts of both SEC62 and SR proteins in cells that were treated with SEC62 siRNA only, data shown in figure number one. So where do my eyes go on the graph? Well, I kind of highlighted there with a little box. For B2, here's my answer. The researchers included both proteins SEC62 and SR to show how SEC62 protein are affected, and not, take a look, at the SR protein content when SEC62 siRNA is added to cells, as what I just explained earlier. All right, so that's the answer. For B3, the question asks, based on figure number one, describe the effect on the production of SR protein when cells are treated with SEC62 siRNA. For B3, here's my answer. Based on figure one, if you take a look at the graph, the levels of SR protein, I boxed it there in red, are not affected when treated with SEC62 siRNA. And if you're wondering why aren't they affected, I said it a little bit earlier. So go ahead and rewind a little bit and you can take a look at my response. The reading continues. These are long FRQs, so you have to approach it with excitement, but also with urgency, because it's 8 questions in about 22 minutes. So you got to keep going. So as I'm reading this, once again, as I'm going over this, this is way slower than how I answered it when I took it for fun, which I really do take these for fun. So let's go ahead and keep reading. FRQ number 1 continues by giving you information saying that the researchers then measured the amount of each of the 3 different proteins that was transported to the ER in cells treated with SEC62 siRNA and SR siRNA. The researchers calculated the percent transported relative to the cells treated with control siRNA. All right, so here is new stuff. It then tells you that there's 3 proteins, 1, 2, and 3. As you can see, here's the data. The C1 continues, asks you, identify the independent variables in the researcher's second experiment, data shown in figure 2. All it is the x-axis. What is on the x-axis? Here's my answer. The independent variable shown is the siRNA added to the cells, either the control SEC62 or SR. That is a very simple answer. C2, here's the question. Based on figure 2, identify the proteins, either 1 or 2 or more, it says proteins, that when treated with SEC62 siRNA showed an increase in percent transport to the ER compared with the control. As you can see, here's my answer. I believe the answer is both proteins 2 and proteins 3. As you can see, as you can compare them right there, showed an increase in percent transport to the ER in comparison to the control. Here, I'm looking at proteins 2 and 3. As you can see, they show an increase here and here, okay? So that's my answer right there. Could be right, could be wrong, but that's what I got. C3 is a very simple question, but you got to go quick, and you got to understand that this is asking you, do you understand how many letters, how many nucleotides are going to be found in an amino acid? And we learned that a codon makes up 1 amino acid, and a codon is made up of 3 nucleotides. So 3 nucleotides equals 1 amino acid. That's what it was trying to ask you if you knew. So here's the question, C3. Protein 1 is encoded by 234 nucleotides, while protein 2 is encoded by 495 nucleotides. Assuming that all nucleotides for both proteins include amino acids, that's the key right there, calculate the difference in the number of amino acids between the 2 proteins. You cannot just go 495 minus 234. That should be like, I don't know, 261. That's wrong. It's asking you how many amino acids are encoded. So if you look at it, here's my work. Protein number 1 is 2, and you do not need to show your work. All you need to do is write your answer. So I did not show my work when I was answering this, which made it a lot quicker. But here's the work, because I want you to know at home kind of how I got the answer. 234 nucleotides divided by 3 bases per amino acid is about 78 amino acids. Protein number 2 is made up of about 165 amino acids. 165 minus 78 is about 87. That's my answer. Don't know if it's right, but pretty sure it's right. But that's what I got. When you take a look at D1, researchers claim that protein 1 is the only tested protein that is transported to the ER following its complete translation in the cytosol, in the cytoplasm. Using data from figure 2, support the researchers' claim. All right, so to kind of go back to, once again, a lot of the times in these FRQs, there's little hints in the question. So you got to read quick, you got to kind of understand what the question asks. So going back a little bit to my notes earlier, when you take a look at, and it's asked here, let me double check, protein 1, ER, figure 2, yes. So when you take a look at this, the SR is necessary for transport during translation, and the SEC62 is necessary for transport after translation. So I remember that in the reading, and that's why my answer is shown as here, D1. Here's my answer. As shown in figure 2, protein 1 transport decreases because of the addition of SEC62 and not SR. Take a look at the graph. The researcher's claim is then further supported because, as I said earlier, SEC62 is necessary for transport after translation. I don't think you need that second sentence, but just in case you do, there's a little bit of support to the researcher's claim. For D2, here's the question. For any protein that enters the ER, researchers claim that amino acids close to the protein's amino acid terminus, the amino terminus, determine how likely the protein is to pass through the protein channel within the ER membrane. Justify the researcher's claim based on your understanding of factors that affect the transport of proteins across membranes. So some quick teaching real quick before I go into the answer. We learned in class that amino acids are all the same, but they're different because of their R group, as you can see down here. But they're the same in that all amino acids have a carbon in the middle, a hydrogen group, an amino group, NH2, and a carboxyl group, COOH, hydroxyl. So here's something else. We also learned in class that where does an amino acid, where does a protein begin? When you look at the first amino acid, specifically its N-terminal. Where does it end? Specifically in an amino acid, the last one, the C-terminal of the carboxyl group, if you look at the end. So here's my answer. For D2, proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. So that's usually what's within a protein. There's a signal sequence close to the amino acid terminus, as I said earlier. Also, I don't know if you need this, but I also added in something called SRP. What is SRP? We learned in class that SRPs are signal recognition particles, which will then bind to an SRP receptor protein on the ER membrane surface for it to pass through. So if you have the key, you can go ahead and enter. And that's earlier what it said when it came to the channel protein. So that was FRQ number 1. I went a lot slower than I actually did when I actually took it within the 90-minute period, but hopefully by slowing it down, you had a better understanding of FRQ number 1. All right, here we go. FRQ number 2. Let's keep going. All right, FRQ number 2 begins by giving you information that many insects rely on something called pheromones. We learned about this in class in, I believe, chapter 51 for animal behavior. Chemical signals that are released by the females to find mating partners. Scientists hypothesize that in a certain type of moth, here we're looking at moths here, the behavior of male moths in response to pheromones is actually regulated by an extracellular signaling molecule, a ligand called 20E. Once again, we've never studied 20E, but you don't even need to know about 20E. All you need to know is that it's a signaling molecule. 2A asks many, okay, so here are my notes: insects, pheromones, females to find mating partners. We're looking at moths. And as I said earlier, behavior, male response, pheromones, regulated in extracellular molecule 20E. So here's the first question. Usually, once again, the first question is very simple, and they want to kind of help you get some points here. For 2A, many receptors are embedded in the plasma membrane. Describe the polarity, polar or nonpolar, of the portion of the receptor that is inside the membrane. In class, we learned that the polarity of the portion of the receptor that is inside the membrane is actually going to be nonpolar. I don't know if you need this additional information, but here's what I wrote. Phospholipid bilayers are semi-permeable because, so why are certain things allowed into the membrane and certain things are not? Why? Because of the phospholipid bilayer, because of the hydrophilic heads that face outside and inside of the cell, and the hydrophobic tails that face inwards. Because of the hydrophobicity and the hydrophobicness of the inside of the cell membrane, that is what allows certain things not to pass. All right? It then continues with some more information for FRQ number 2. To investigate whether the binding of 20E to its receptor, DOPECR, we have not learned that. All you need to know is that it's a receptor. Affects behavior in moths, scientists injected male moths with saline or control solution. Or, once again, here it comes up again, but once again, you don't need to know too much about this other than what it's telling you here. Or with something called small interfering RNAs, siRNAs molecules, that inhibit the expression of the gene encoding DOPECR. The scientists then exposed the moths to the pheromone and determined the percent of total time observed that the moths engaged in general activity. Okay, there's general activity, and then there's something later on called oriented activity, which I'm going to keep reading here. Defined as movement in any direction. The scientists all determined that the percent of general activity time that the moths spent in oriented activity defined as the movement towards an area of high pheromone concentration. What is the difference between those? I wrote it down here, and here I just took some quick notes as I was writing down some information. Typically, I don't reread FRQs twice. Why do I take little mini notes as I read? So that I can just look at my notes and I don't have to reread it. Sometimes I go back and look at information, but that takes time. So if you're rereading this 5 times, you're probably not going to finish. And it's all a time test. It then gives you table number 1, which I quickly looked at it when I took it, but because I'm reviewing this with you, I'm going to quickly explain a little bit more. Table 1, we look at 2 different treatments: male moths that are injected with saline or controlled solution. And then we've got a treatment of male moths injected with siRNAs that inhibit or stop expression of that gene, encoding that receptor, DOPECR. Here we see the general activity and the oriented activity, and we see some numbers. We see, so real quick, as I'm taking a look at this information, I'm looking at how for general activity, it's about 95 and 90, okay, pretty similar. But then I see a big red flag or something that stands out. For oriented activity, we see 60 and 25. That's a big difference versus here, we didn't see much of a difference. So that's all what's going on in my head as I'm reading this question. As we continue reading, it gives even more. It's a really long question. DOPECR is a G protein coupled receptor. When 20E binds to DOPECR, GTP displaces GDP bound to the G protein, and a signaling pathway is activated. The scientists hypothesize that this leads to the transcription of genes associated with the oriented activity observed in the male mouse. Okay, so here we see GPCR, which is one of the three receptors that we study, GPCRs or RTKs, or the ligated ion channels, when it looks at the cell membrane receptors. But here I just wrote down that 20E is going to bind, and that's going to ultimately cause what? Transcription of genes within oriented activity. This is the picture that's shown here. We have not seen it in class, but we've seen something very similar. And I'm going to quickly go to what we've seen in class. In class, in chapter 11, we studied cell communication. This is not a figure given on an AP exam, but it's a picture that was studied in class. And what did we learn? G protein gets activated. It activates an enzyme called adenylyl cyclase, activating a secondary messenger, CAMP. And ultimately, it's going to lead to a cellular response. So how does this relate? It's actually pretty similar. Because here it just tells you that when the G protein is inactive, here's what's going to happen. But on the right, when the G protein is active, it's going to lead to what? It tells you an increased expression of genes associated with oriented activity in male mouse. And this is once again on the cell membrane. So it asks you, using the template in the space provided for your response, construct an appropriate type of graph that represents data in table 1. So here we go to table 1. Here's what we're looking at. When I graphed this, I saw that it was a comparison between the two different treatments. I saw general activity and oriented activity, and here's what I saw. So I'm going to go back here again. 95 plus or minus 5. If you don't know how to do error bars, the quick way to do it is first you would graph 95, and here you would graph 60. But then the error bar is plus or minus 5, so we would go up to 95 to 100, and then you would go down from 95 minus 5, which would be to 90. So what do error bars kind of tell you? They tell you if whether or not the data is statistically significant or not. So you can look more into that if you need to. But here we quickly see that for saline control, general versus oriented, and then we see for siRNAs for general versus oriented, we see that, once again, there's a big difference. General is a lot higher than oriented, which is a lot lower. Now, none of the error bars will overlap, which means what? And the difference will mean that the means, as shown here, the numbers are statistically significant, which means that we can take them as we can see them. Okay, so for B2, we take a look at how the question asks us, based on the data in table 1, determine the type of activity that was affected by inhibiting the expression of the DOPECR receptor. So here's my answer for B2. By inhibiting the expression of DOPECR receptor, what's going to happen? The oriented activity, as shown here in the data table, was actually affected the most. Okay, so as you can see, the inhibiting the expression of DOPECR, right here, we see the oriented activity was affected the most. So that's pretty straightforward. For C1, it asks you, based on table number 1, identify the treatment group in which the oriented activity was greater than 50% of the general activity. So as you can see here, the general activity is shown here, 95 and 90. What's 50% of 95? Roughly about 47.5, something like that. So which oriented activity is greater than 50% of that general activity? Well, 60 is greater than 47.5 versus 90, 50% would be about 45. 25 is less than. So the answer is the saline control, as you can see shown here, showed oriented activity greater than 50% of the general activity. That's what I got. FRQ2 for C asks you, the scientists studied some moths with a mutation in the gene encoding the G protein. The mutation prevents GTP from displacing GDP bound to the G protein. Based on figure 1, predict the effect of this mutation on the oriented activity in male moths exposed to the pheromone. Okay, so here, once again, we're going back. Look at what the image told you. It showed you that when GTP when G protein's activated, there's an increased expression of genes associated with oriented activity in male moths. Okay, so here's my answer. C2, this mutation will actually reduce the activity because you're mutating and you're not allowing what I just said right here, It's going to reduce the oriented activity in male moths exposed to the pheromone, okay? Because that's just what the mutation is. That's my answer. It gives you more, but wait, there's more. FRQ number 2. When we look at the reading, it tells you that the expression of the gene encoding DOP-ECR is low in male moths during when? The first few days as adults, when they are sexually immature. Gene expression will rapidly increase as the moths reach sexual maturity. The scientists claim, here's the claim, that this increase in gene expression increases the likelihood of males finding females with whom to mate. So here's some quick notes that I took down, and as I wrote down some. Now, once again, because of time, I don't actually write this out, but I'm making little notes on the side, pretty much telling me that the expression of gene is low in males in the first few days, but then it rapidly increases at sexual maturity. And then once again, I just quickly wrote down the claim, because we're going to actually need that. Here's the question here. Use evidence from the information provided, which I just read to you, to provided to support the scientist's claim. Once again, what's the claim? That an increase in gene expression will help males finding mates. Here's my answer for D1. The expression of the gene encoding DOP-ECR, this receptor, rapidly increases as the moths reach sexual maturity, which supports the claim that this increase in gene expression will increase the likelihood of males finding females to mate with. That was my answer for D1. But here's D2. Based on figure 1, explain how an inhibitor of, if you were to stop it, of the DOP-ECR pathway might serve as an effective chemical to protect crops from moth damage. So when you take a look at it, if you're inhibiting the DOP-ECR pathway, here's my answer that I wrote out. D2, as shown in figure 1 in this chart right here, when the DOP-ECR pathway is inhibited, activity in males, take a look, is lowered. And thus mating, which leads to reproduction. If you think about it, male and female mating will lead to reproduction of more moths, will be lowered as well. The lower moth population will ultimately help protect crops. It's asking you explain how it's going to help protect crops from moth damage, because I would think that more moths would create and more would lead to more moth damage. All right, that was really long, but when I took it, I was watching my time and I realized I was going too slow. And even me, I've taken so many of these, I was going too slow. So you got to keep moving. So as I'm taking it, I was watching the time and I took about 25 minutes each on 1 and 2. So that took about 50 minutes of the 90, which gave me about 10 minutes on each of the last 3 FRQs, which are worth about 50% of the score, including time to read it. So the next 4 are pretty quick, except FRQ number 6. When I got to it, I had about 8-9 minutes left, and I realized I should have gone faster. So if you felt the same when you took it, I get it. I got it. I get it. So here's FRQ number 3. Buffalo grass is an invasive species. Here, I just took some notes. I just put BG invasive. That's all I put when I was writing. Invasive to the what? So it's in desert ecosystems, and it's threatening something called saguaro cactus, which is a keystone species. We learned about keystone species in class. Now, this buffalo grass is also drought tolerant versus, and it can survive sorry, I'm supposed to be reading it. And it can survive wildfires. However, the dry buffalo grass also acts as fuel for fires, so it can increase wildfires, causing the fires to be more severe. Other saguaro cacti can survive wildfires. However, many of the young cacti cannot. So I just quickly took some notes down here as I'm reading it one time. It then goes ahead and tells you that scientists conducted an experiment to determine whether they could control the abundance of the buffalo grass population. The scientists identified several native grass species that, when grown with buffalo grass, might reduce the abundance of buffalo grass. They grew buffalo grass in the presence of several different native grass species in greenhouses in either non-drought, watered every 3 days, or drought, watered every 9 days conditions. After 12 weeks, they measured the dependent variable, the height and dry weight of the buffalo grass in each treatment group. So I just took some notes right there, and hopefully that helps out. So here's the first question, 3A. Describe the effect of removing a keystone species that will have on an ecosystem. You don't even need to know about buffalo grass, but here's what you should have known about keystone species. 3A, a keystone species maintains an ecosystem's balance, stability, and diversity. We know that it keeps everything in check. If you remove it, everything goes out of check. So by removing a keystone species in general in any food chain, an ecosystem will not be balanced, stable, or have that diversity that we want when it comes to species diversity. 3B, Here's the question. Identify a control group that scientists should include in their experiment. If you go back to the reading, it says that they're going to use several different native grass species in non-drought or drought conditions. So here's my answer, 3B. Scientists should include a control group of what? Buffalo grass that is not grown with any native grass. So just buffalo grass species in both non-drought and drought conditions. 3C, state the null hypothesis of the experiment in which the buffalo grass is grown in the presence of native grass species. We practice this a lot in class. What is the null hypothesis? It's not just an educated guess, but it's usually something that goes along with this statement right here. Native grass species have no effect on the height and dry weight of buffalo grass. Any single time you write a null hypothesis, you're pretty much saying that there's no effects. If you're ever asked, which you weren't asked in this question, but if you're ever asked about an alternative hypothesis, as we practice in class, if it asks for that, here's what the alternative hypothesis would be for the same question. You would just write, native grass species will have an effect. It's the opposite of the null hypothesis. It would have an effect on the height and dry weight of buffalo grass. 3D, scientists have found that the population growth rates of native grasses are much slower than the population growth rate of buffalo grass following a wildfire. I just told you right there. The scientists claim that wildfires will therefore increase the abundance of buffalo grass plants in an ecosystem. Based on the information given, justify the scientists' claim. So here's my answer for 3D. Buffalo grass plants, as it said in the question, have a faster growth rate in following a wildfire versus native grasses. After a wildfire, there will be more buffalo grass plants in the ecosystem. That one was interesting. I pretty much just restated what the question said. So that's me justifying the scientist's claim. That's what I got. All right, here we go. FRQ number 4. All right. FRQ number 4. Let's read it together. 20 million years ago, the Caribbean Sea, I just think about the Caribbean, kind of like below Florida, to the side of Florida, the Caribbean islands, and the Pacific Ocean, which I think is on the west coast of North America and South America. Pacific Ocean were connected. So that's what was shown here in the question. And water flowed freely between the two bodies of water. Okay. Many of the same marine species were found in both areas. Over millions of years, the land referred to as Isthmus of Panama formed. We've never learned this in class. You just kind of look at what the question gives you. Eventually closing off the connection between the Caribbean Sea and the Pacific Ocean, creating two separate bodies of water, now as we know as the Pacific and let's say the Atlantic Oceans. The ecology of these two marine habits was dramatically altered by this land formation. The warmer Caribbean water, as we know on the east of North America, could no longer flow west. So the Pacific Ocean cooled and became more nutrient rich, which is what we know, while the Caribbean water became warmer. 4A, describe the genetic evidence that evolution is occurring in a population. You don't even need to know the question. You could just answer this based on what we learned in biology. Here's my answer. So as we learned in class, evolution occurs in a population when there are changes in allele frequencies over time. You don't need to go into it, but that's why we have Hardy-Weinberg. Ultimately, what we're doing is we're comparing before and after these allele frequencies. And if there's a change, then there will be evolution. If there's no change, then the population is in genetic equilibrium, and thus it's not going to have evolution. All right, we learned that in class. 4B, explain how the isolation of marine species by the formation of a land barrier can lead to divergent evolution. So species becoming different over time of those species. Here's my answer. 4B. Speciation, as we know, and divergent evolution take place when marine species, in this case, are separated or isolated for a long period of time, and reproductive isolation is achieved. So I continued writing, a new species can then form when reproductive isolation prevents gene flow between marine species populations due to a land barrier. Okay. That's what I got. We'll take a look at the answer key when it comes out. So we'll compare that. Before C, it asks you predict the effect of the formation of the isthmus had on resource availability for South American species. Here's my answer for C. The establishment of the Isthmus of Panama connecting North and South America most likely will increase competition between land organisms. Think about it. You were separate, now you're together. Most likely, so as it goes on here, justify your prediction. Most likely when connected, the organisms from North America and South America, which were previously had their own separate space, food source, and roles and niches within the community and favorable traits, then they had to compete now against other species. There may be another answer, but that's just what I got, and we'll take a look at it. All right, here we go. FRQ number 5. Let's finish strong. it tells you in this question, it shows you that figure 1 shows the reactions of the metabolic pathway used to synthesize amino acid B from amino acid A in cells. We've never seen this diagram, but you got to read it. Looks like amino acid A is like a ligand that binds to enzyme 1. Enzyme 1 will then lead to something called intermediate X. That will then bind to enzyme 2, which will then lead to intermediate Y, which will then bind to enzyme 3. Woo, it's a lot going on, which will ultimately lead to amino acid B. It looks like there's an arrow that goes back. And in class, we learned that this is something called feedback inhibition, and that pretty much this and it's going to inhibit enzyme 1 in this case, which will then prevent overproduction by regulating inhibiting enzyme 1. Okay, so it all goes back. Okay, so you can see that. So that's all what's going on in my head. I'm writing down some quick notes, but that's what I'm trying to show you what's going on in my mind. 5A, finish strong. Describe a characteristic of an enzyme's active site that allows it to catalyze a specific chemical reaction. Once again, you don't even need to know anything about the question. You just need to know that shape determines function. The shape of an enzyme's active site will determine its function, and specificity as to which substrate will be able to bind and which chemical reaction can take place. For 5B, based on figure 1, explain how the binding of amino acid A to enzyme 1 is regulated by amino acid B. So here's my answer. According to figure 1, the concentration of amino acid B determines the concentration of amino acid A due to what? Feedback inhibition. I said that a little bit earlier, helping maintain the concentration of the desired level. For FRQ5C, here's the question. Using the information in figure 1, identify the product of the reaction catalyzed by enzyme 2. Is it going to be intermediate X, Y, or amino acid B? Pretty straightforward here. I did it very quickly. 5C, according to figure 1, the product of the reaction catalyzed by enzyme 2 is going to be intermediate Y, shown right there on the picture. D, 5D, based on figure 1, explain how a change in pH could affect enzyme 3 in such a way that amino acid B cannot be produced. Okay, here's my answer, 5D. Sorry, that should be 5D. Enzyme 3 can lose its shape and function. We've learned that enzymes can be denatured through pH, salt concentration, or temperature. Because of denaturation due to an environmental change in pH, thus stopping, if you mess up its shape, you're going to mess up its function, stopping production of amino acid B. Okay, last one, everybody, let's go. FRQC 6. And this one, I got to tell you, I had about 8-9 minutes from what I remember when I was looking at the timer. This one drove me crazy because I looked at it and I was like, whoa, it's so long. So it's one of those things where don't let it scare you. Look at what it's asking, quickly read it, and answer the question. Here we go. 6. The ALD gene of fruit flies encodes the ALD protein. Genes turn into protein, which is associated in both the centromeres of chromosomes and protein filaments produced during meiosis. Took some quick notes down here. In the absence of functional ALD proteins, gamete-producing cells enter anaphase 1 before homologous chromosomes are correctly aligned. As a result, these gametes produced will not contain the correct number of chromosomes. All right, so that's what we're looking at. It then tells you that scientists generated 4 mutations. We've never learned this in class. You just kind of read it and quickly look at it and understand it. ALD1, 3, 23, and DEL, which was a deletion of the gene. To study the role of ALD protein in meiosis, scientists used gamete-forming metaphase cells from groups of flies with different ALD genotypes. Some of the flies were homozygous with the wild-type allele, okay, WT, homozygous, while other flies were heterozygous. So there's 4 of them. WT-del, ALD1. Okay, so look, there's all 4 of them. Once again, I'm not really thinking too much about writing these down. I'm just keeping in mind that these are heterozygous. The scientists measured the percentage of metaphase cells that contain ALD-associated filaments and the amount of ALD protein produced by each of the cell types. And then it shows you this. And quickly I looked at it and I looked at how it's measured. The percent of metaphase cells with ALD-associated filaments. Here we see WT, that's the homozygous, and these are the heterozygous. So already in my mind, I'm looking at, okay, heterozygous looks higher than most of it, except this one. This heterozygous WT-del is showing a higher percent of metaphase cells with the ALD-associated microfilaments or filaments. Figure 1B showed you that, look, this homozygous has a bigger, and it tells you right here, a thicker band indicates a greater amount of ALD protein. So more protein for homozygous, less, about half for the heterozygous, about the same amount, and then less and nothing for this ALD23-del. So for 6A, it asks you, based on figure 1A, as identify the fly genotype in which the average percent of metaphase cells with ALD-associated filaments is close to 12%. Here's 60%, which one's close to 12%? I just quickly took a look at it right here. And here we look at fly genotype ALD1. You don't even need to know anything about question. You just got to read a graph. Has the average amount of metaphase closest to 12%, in my opinion. Okay. 6B, based on figure 1B, describe the difference in ALD protein production between the gamete-forming metaphase cells of flies with the genotype, right, ALD 323, flies, etc. Here's my answer. 6B, according to figure 1B, take a look. Gamete-forming metaphase cells of flies with genotype ALD3, 23 will produce more ALD protein than flies with this. There's none. Okay, so that's all you need to know for B. So pretty straightforward. For C, 6C, scientists hypothesize that gamete-forming metaphase cells can produce a normal amount of ALD-associated filaments even when they produce about half as much ALD protein as the wild-type cells produce. Use the data in figures 1A and B to support the scientists' hypothesis. Here's my answer, 6C. According to the figure 1A, we see that WT-del has a greater percent of ALD-associated filaments than WT-WT. However, look over here. In figure 1B, we see that it produces about half the amount as the ALD protein. I say that this data shows that half the amount of ALD protein is actually enough to receive a normal amount of ALD-associated filaments. Last one, everybody, 6D. For gamete-forming metaphase cells of WT-del and ALD1 del flies, explain why the phenotypes observed in figure 1A, why do they differ, even though the amount of ALD protein produced does not? And here is my answer for 6D. The amount of ALD protein produced shown in WT-del and ALD1-del flies is actually pretty similar. But what we see a difference here is, so here's my answer. But the phenotypes in figure 1A differ because of ALD1 allele, most likely. Okay, so something kind of went wrong if you think about it. Has a different non-coding, let's just say gene, than WT that will produce, let's just say, a lower amount of ALD associated filaments. So this one, that's just what I got. I could see another answer, but let's just see the answer key when it comes out. All right, here we go. Thank you, everybody. Man, if you watched this till the end, hopefully it helped. Whether or not you took it already, it's already done. So don't stress about it. But if you haven't taken it, hopefully this helps for preparation. That timing is huge. So go quick and make sure you practice. This is as much as you can, as many practices as you can. I hope this is helpful for you. I wish you the best on your AP tests. If you already took it, I wish you the best on your next AP test. Remember, I truly believe that you are not defined by your AP score. I truly believe that because you challenge yourself by taking AP Bio or this AP Bio exam, you're going to learn so much from it. Even if you don't do well on the exam, you're going to learn so much. Everyone, and I really believe this, and you need to hear this, no matter what you get on this AP exam, I believe that everyone, if you keep working, if you keep having delayed gratification and perseverance, you will have a great future ahead. So keep enjoying bio and keep practicing. And this is just one test in so many in your life that you'll have many more chances if you don't do well. So reflect, grow, and continue enjoy learning. I'll see you next year. And once again, I really enjoy taking these exams, and I hope that this was helpful to you. Thank you, everybody. Have a good one.